Dr. Stephanie Seneff. Dr. Stephanie Seneff, author of The Toxic Legacy of Glyphosate. She's a senior research fellow at MIT. For over 3 decades, her research interests have always been at the intersection of biology and computation, developing a computational model for human auditory system, understanding human language, applying natural language processing techniques to gene predictions. In the recent years, Dr. Seneff has focused her research interest back towards biology. She's concentrating mainly on the relationship between nutrition and health. Since 2011, she has published over 2 dozen papers in various medical and health related journals on the topics such as modern day diseases like Alzheimer, autism, cardiovascular diseases, analysis and search of databases of drug side effects using NLP techniques, and the impact of nutritional deficiencies and environmental toxins on human health. Today's talk, what Stephanie is going to talk about is on long COVID, neurological aspects, role of senescence, and the treatment options. So without any delay, I would like to invite Dr. Seneff.
Hello, everybody. Thank you for being here. Can you see my slides? Am I good?
Yes, it's absolutely great. Thank you.
Okay, good. Thanks. Just wasn't sure. So long COVID, neurological aspects, the role of senescence and treatment options. And I want to say first, thank you for talking about my work on glyphosate, my book, Toxic Legacy. I noticed both of the speakers in passing mentioned organic food or glyphosate. I think it's a really important aspect of long COVID to make sure that you eat a certified organic diet. I think it may be one of the most important things you can do to protect yourself from long COVID and to treat long COVID. Stay away from the toxic chemicals in the food. You know, the water, the air, those are problems too, but the food is very, very important, and especially the glyphosate. I won't be talking at all about glyphosate in this presentation, however. So here's an outline of my talk. First, what is long COVID? Second, neurodegenerative disease. The third topic, very, very interesting to me, is syncytia and senescence. And then finally, natural treatment options and a summary. So people have sort of assumed that everybody knows what long COVID is, and I'm sure this audience probably does. But some information from the CDC, it can last for weeks or months or even years. It's found more often in people who had a severe case of COVID. The symptoms often resemble myalgic encephalomyelitis. It's a big word, myalgic encephalomyelitis, or chronic fatigue syndrome. And so some of the common symptoms include this post-exertional malaise, exhaustion, heart palpitations, headache, brain fog, lightheadedness, depression, anxiety, joint muscle pain, diarrhea, lots of problems. Fatigue being the most overwhelming problem that people face, fatigue and brain fog. And then here's another poster from the CDC showing approximately 1 in 5 adults, which was shocking to me. 20% of people who get COVID have long-term consequences over the age of 18. And these consequences are can cover all the many different aspects of the body, cardiovascular conditions, kidney failure, respiratory problems, musculoskeletal, blood clots, and of course, the neurological and mental aspect, which is what I'll be talking about today. So what causes long COVID? This is still, I think, more or less a mystery, although there's a lot of hints that it's coming out of the gut, and certainly from the previous 2 speakers. And I would agree with that. I think that's where it's being held. The virus is surviving in the gut, or just the proteins are surviving in the gut, not clear. But that's maintaining an inflammatory state in the gut. And then that causes mechanisms that cause the virus to spread. So this is where I'm going to go into this issue of syncytia, where the virus is able to spread from cell to cell through a novel mechanism involving syncytia, where multiple cells merge together into a giant cell. And then the spike protein is extremely interesting, acting as a prion-like protein. It can penetrate the brain barrier and it can induce neurodegenerative disease. So that's what's really scary about this virus. That protein is very toxic. And the virus actually induces a state of senescence in the immune cells or even in tissue cells, the neurons, the fibroblasts, et cetera, the heart, cardiovascular cells. So these can cause these cells to be sick. So just starting with neurodegenerative disease, can SARS-CoV-2 productively infect the upper or lower gastrointestinal tract? I've seen from my previous speakers that they have shown that this is very clear. and even whether it might be acting as a bacteriophage and infecting the bacteria in the gut. Very, very interesting question. But it seems that the virus can be sustained in the gut for a long time after the person has recovered, or at least the proteins that are produced by the virus are somehow being held by some certain cells and kept alive. And so those proteins are causing these responses. There's a high rate of fecal RNA shedding. This was talked about by the other presenters. And viral RNA has been detected in these fecal samples. And so we need further study to figure out exactly what's going on in the gut. But it is clear that that's where it feels like the bacteria are helping, collaborating with the virus to keep it alive in the gut. So this is an interesting paper from 2021, Parkinson's disease and post-COVID-19 syndrome, the Parkinson's long COVID spectrum. And they looked at 27 patients who had Parkinson's disease and then caught COVID-19 across several centers. And they found lots of evidence of post-COVID-19 syndrome. 85% of these patients continued to suffer after they had recovered from the disease. And they had lots of worsening of the various functions that are associated with Parkinson's disease. The motor function, they needed more L-DOPA to maintain their ability to move. They had increased fatigue and cognitive disturbances. 22% had cognitive disturbances and sleep disturbances, but 85% had long COVID, and then you have half of them with these other problems. So clearly a bad effect of COVID-19 on Parkinson's patients. Now, this paper, I think, is very interesting. It's from 2013, so it has nothing to do with COVID-19, but it really paints the picture of what I think might be going on in the case of long COVID and how it can cause Parkinson-like symptoms in the brain. And so, first of all, there are terminals from the dorsal motor nucleus of the vagus nerve that sit within the gastric submucosa just a few millimeters away from the lumen. So very close contact between the gut and the vagus nerve. And now exosomes are a really important part of the process. These are lipid particles that are released by cells under stress, especially by the immune cells under stress, and they can contain the spike protein itself, because these immune cells that are getting infected or getting exposed to the virus, they pick up the spike protein and they release it in the form of exosomes. And those exosomes can then migrate to the brain and cause problems. They're delivering the spike protein to the brain. They also contain alpha-synuclein, which is upregulated in response to the virus. And then they can become toxic to neurons. This has been shown in studies that they're toxic to cultured neurons, the exosomes that are produced by the immune cells that are infected with the virus. So alpha-synuclein migrates from the gut and from the nose to specific regions of the central nervous system, and there they can cause then these problems. The earliest symptoms, when you're looking at Parkinson's disease patient, early on, before they've even expressed symptoms, they've got problems with the smelling. The sense of smell starts to fade, and they're having, and the dorsal motor nucleus of the vagus nerve those are the early indicators where Parkinson's seems to first start, with this alpha-synuclein misfolding. And then aggravated alpha-synuclein from the affected neurons can be transferred to unaffected neurons via these exosomes. So the exosomes allow the immune cells to deliver the toxic alpha-synuclein along with misfolded foreign proteins such as the spike protein. Excuse me. And then they can transfer from the immune cells to the neurons and from the neurons to more neurons. So this is really a nasty consequence. Now, there's papers that are showing that SARS-CoV-2 proteins are, in fact, prion-like, and they can be the protein, the external protein that you're exposed to, that can cause this whole process that I just talked about in the previous slide. SARS-CoV-2 proteins interact with alpha-synuclein, and they induce these Lewy bodies, which are the characteristic feature, these precipitates that contain the alpha-synuclein, the spike protein, the proteins in the virus can cause this to happen. And so alpha-synuclein is upregulated in cells that are exposed to the virus, and then it gets aggregated. It's accelerated aggregation as a consequence of both the S protein and the N protein, the nucleocapsid protein. And then they showed that these proteins cause Lewy-like pathology in the presence of excess alpha-synuclein. And then by confirming that SARS-CoV-2 proteins directly interact with alpha-synuclein, our study offers new insights into the mechanism underlying the development of Parkinson's disease on the background of COVID-19. And I should point out that this spike protein is quite interesting. It has 7 of these classic glycine zipper motifs that are characteristic of prion proteins. And there are 7 of them in this protein, which is quite unusual. So now this slide, this is an interesting paper here, interactions between SARS-CoV-2 N protein and alpha-synuclein accelerate amyloid formation. So it's the same message again. The nucleocapsid protein speeds up the aggregation. Multi-protein complexes form amyloid beta plaque. And our results point toward direct interactions between the N protein of SARS-CoV-2 and alpha-synuclein as a molecular basis for the observed correlation between SARS-CoV-2 and Parkinsonism. So now my next topic is syncytia and senescence. Senescence-associated inflammatory responses, aging and cancer perspectives. Now this is, again, a paper from pre-COVID paper 2015 talking about senescence, and it's a very interesting cellular response to injury. And it results in a permanent arrest of the cell cycle. So the cell sits there, but it doesn't die. This is the critical thing with senescence. And it sits there and it is metabolically active, and it produces these cytokines and chemokines, various kinds of chemicals. It's called senescence-associated secretory phenotype. This phenotype of the cell causes it to release metabolically. It's metabolically active and causes it to release a lot of proteins that exert a paracrine effect on other cells. So it's putting these out into the environment. The other cells are picking them up, and then they can also respond by becoming inflammatory and also becoming senescent. They link senescence to inflammation. These things that they release link it to inflammation. This is a very, very fascinating paper to me that I found only recently, a very new paper, 2021, micronucleus production, activation of DNA damage response, and CGAS sting signaling in syncytia induced by SARS-CoV-2 infection. And they basically looked at these HeLa cells that had been engineered to have ACE2 receptors. They don't normally have that. And they were grown in culture. And then they exposed them to the spike glycoprotein. And ACE2 receptors allowed them to take it up. And they formed these multinucleated syncytia. And this is a schematic of it, two cells coming together, multiple cells ending up with a single giant cell with multiple nuclei, and also with these micronuclei, which are an indicator of DNA damage, DNA double-strand breaks. And that, of course, can induce serious problems like cancer. So the syncytia succumb to DNA damage in response to this to infection with the virus. And that leads to genomic instability, causing these micronuclei to appear. And then the CGAS sting signaling pathway is a characteristic feature of this senescence-associated sensory phenotype. So this is another paper that talks about infection spread through cell-cell contact. And this can be because these cells blend their boundaries and are able to just push the protein right over without ever going into the extracellular space. So this sort of evades neutralizing antibodies because the cell's just giving it to the other cell directly. And so the syncytia formation is a way to do that. Now, all these cells come together, they all get sick with the virus. And they can also form these really fascinating, they send out these filopodia, which eventually can become tunneling nanotubes. And this cell can just deliver the virus and lots of other things as well to the recipient cell. SARS-CoV-2 infects cardiomyocytes and induces syncytia formation. This is a fabulous paper, and it has some beautiful figures in there. I show one of them here with these filopodia linking these two cardiomyocytes. So these are heart muscle cells that are blending together connecting with each other through these filopodia in response to the SARS-CoV-2 infection. And so, in fact, these muscle cells in the heart contain abundant levels of ACE2, so they very readily get infected by the virus if it gets into the heart. And then they noticed that the viral transcripts accounted for 88% of the total mRNA being produced by these infected cells. So they were just focused on making virus. And the particles ended up covering the cell surface of these cells. Fabulous pictures in this paper. And then the spike protein induced this filopodia formation, and then it caused fusing of the cardiomyocytes, generating these syncytia. And so furin cleavage, actually the furin cleavage site, which is a unique feature of SARS-CoV-2 compared to SARS-CoV, that was essential for the syncytia formation. You needed to cut the spike protein in half into S1 and S2. And then there were abundant progeny virions that were found in these exosomes that were released by these cells. So here's another paper. Post-acute COVID-19 is characterized by gut viral antigen persistence in inflammatory bowel diseases. And others in this conference have talked about this already, but it's quite fascinating. The RNA was expressed in the gut mucosa 7 months after acute COVID in 32 of 46 patients who had IBD. And the nucleocapsid protein was also detectable in 24 out of the 46 patients in the epithelium of the gut and also in the CD8 plus T cells, the immune cells. And then the antigen persistence was not obviously related to the severity of the original disease. But when they looked at the post-acute sequelae, the long COVID conditions, they were predominantly present in the patients who had persistence of the viral antigen. So it looks like the viral antigen is a causal contributor to long COVID, but they didn't prove that. So whether viral antigen persistence in and beyond the gut underlies the pathophysiology of post-acute COVID warrants further clinical trials. Here's a paper from 2022, the role of senescence in the chronic health consequences of COVID-19, innate immune system dysfunction that leads to decreased senescent cell removal or increased senescent cell formation could contribute to accumulation of senescent cells with both aging. That sort of senescence is a characteristic of aging and also with viral infections. And so it could be that the senescent cells, many are being created, or it could be the ones that are created not being removed by the immune system because the immune system is defective. Both of those are probably going on. And this is just a nice picture of the lungs infected with the virus, young versus old. So in the old lungs, you have a lot more immunosenescent cells showing up here. These guys down here, lots of different types of immunosenescent cells. They're surviving, but just surviving and releasing chemicals that are going to cause trouble, inflammation. And so when you're old and you have a lot of those, you have a much more severe case, typically of COVID-19, and more inflammation because of the release of the cytokines and chemokines by the senescent cells. And virus-induced senescence is a driver and therapeutic target in COVID-19. So SARS-CoV-2 invokes a cellular senescence response in infected cells, which is accompanied by this senescence-associated secretory phenotype. It includes pro-inflammatory cytokines, extracellular matrix active factors, things that can break down the extracellular matrix, and also procoagulatory mediators that can cause blood clots. And then, interestingly enough, you can get secondary senescence of endothelial cells. It might not affect the virus, but they can pick up those secretions and end up also converting into senescent cells. And then the supernatant from those infected cells induce platelet activation and the clotting cascade. So again, those signals that are being released by those senescent cells are causing a lot of trouble. SARS-CoV-2 infection causes dopaminergic neuron senescence. So now we're getting into the nervous system. And this paper shows that infection in the neurons in the substantia nigra that are associated with defects are associated with Parkinson's disease. And when they're infected with SARS-CoV-2, an inflammatory response and the cellular senescence response are apparent. And these cells do express ACE2, and so that's going to promote viral proliferation inside these cells. And then they used a high throughput screen to identify several FDA-approved drugs that could help to rescue the cellular senescence phenotype. Riluzole, metformin, and imatinib were drugs that they found could be useful. And now, so this is really kind of a summary that brings together the problems with the problems of the cellular senescence. I think that it all comes together in the long COVID neurological symptoms. You start with the misfolded proteins, and then their recognition as non-self proteins activates the innate immune system. And now that induces inflammation, inflammation induces cellular senescence, and cellular senescence induces more inflammation in a kind of a feedback loop. And then finally, the neurons get injured by this, and you get brain organ failure. Natural treatment options. Emerging senolytic agents derived from natural products. So there's several papers that talk about senolytics. I love that word because these are small molecules that can actively kill senescent cells. And this is what you need. You need to remove these cells. They shouldn't be alive. And so many things that we've heard about already, quercetin, that is a flavonoid in various vegetables, as well as in tea and red wine. Fisetin, which is a flavonoid in many fruits and vegetables, apples, grapes, persimmons, curcumin, which is a polyphenol in turmeric, and then piperlongumine, which is an active alkaloid found in peppers. So these are kind of the one fruits that you can eat and remove cells. So this is a paper specifically on the senolytic effects of quercetin. It means it can kill the senescent cell. fat cells were exposed to reactive oxygen species, and they showed that caused them to move towards a senescent state with these markers like senescence-associated beta-galactosidase activity, increased pro-inflammatory responses, and synthesis of MIR155, which is an inflammatory microRNA. And quercetin improved all of these markers. Many natural compounds have senolytic effects, and here's again many that have been mentioned already: phenols like curcumin and all of these things, organosulfur compounds, sulforaphane, which is found in broccoli and all cruciferous vegetables, methyl tocols, tocotrienols, alkaloids like berberine, and piperlongumine, which was mentioned earlier, terpenoids like triptolide. So all of these good things are going to help to treat this treat these senescent cells. And then here's a paper on Bicalin showing that it actually helps to deal with prion protein misfolding, which is quite interesting. And so junk signaling is a key modulator in cell death induced by the reactive oxygen species. This Bicalin, it's the active ingredient in the dried root of a plant that's known as Huang Qin in traditional Chinese medicine. I think it's a very popular Chinese medicine treatment. It protects the neuronal cells against the prion protein-induced neurotoxicity by inhibiting this J and K activation. And it reduces prion protein-induced mitochondrial dysfunction and also reduces ROS. So this seems like a good option for treatment. And now I want to have a few slides here on melatonin. We'll go through them quickly, but melatonin highlighting its use as a potential treatment for SARS-CoV-2 infection. It looks like there's nothing not to like about melatonin. There's more than 140 scientific publications that have identified melatonin as a likely useful agent to treat COVID-19. It has a high safety profile over a wide range of doses. It doesn't have significant toxicity. It's a confirmed potent anti-inflammatory agent, and it's an antioxidant in many experimental models and clinical situations. And it's also a well-recognized suppressor of HIF-1 alpha, which is what is induced in response to hypoxia. And so here's another paper specifically addressing senescence. Melatonin suppresses senescence-derived mitochondrial dysfunction in these mesenchymal stem cells. So these senescent cells, of course, are generating reactive oxygen species, resulting in mitochondrial dysfunction. Melatonin can prevent the induction of senescence, and it enhances the therapeutic potential of these mesenchymal stem cells, help to keep them healthy. And it activates mitophagy so that you can clear the damaged mitochondria. So all of this is good news. And this is just a graph from a slide in a paper, another paper that talks about melatonin's many beneficial effects, and seeing all these different things that are suppressed by melatonin that are good things to suppress to try to tame the inflammation, to prevent the Warburg metabolism effect that's associated with cancer, where they're going into glycolysis instead of using their mitochondria to make the ATP. It can cause the M1 cells to mature into M2 cells, which are anti-inflammatory, and the protecting from the reactive oxygen species and toning down the inflammation. And so in summary, long COVID is a debilitating disease lasting for months. It could be due to persistent viral infection in the gut long after the acute phase. SARS-CoV-2 spike protein has prion-like properties, and this may be a factor in the neurological symptoms of COVID. SARS-CoV-2 infection is cell-cell fusion, leading to synthetic formation and the evidence of DNA damage and cellular senescence. And many natural molecules, including phenols, alkaloids, terpenoids, organosulfur compounds, have synthetic properties. So that's good news because it needs to treat. And melatonin is a safe and inexpensive supplement exhibiting many beneficial effects, including antioxidant capabilities and clearance of damaged mitochondria. And thank you very much for your time.
Thank you. Thank you so much. Are you there, Dr. Seneff?
I am, yes.
Yes,
Can you
sorry.
hear me?
It was breaking in between. All good. Thank you so much. That was quite insightful. I would just like to know a little bit about melatonin first, because I can understand that melatonin can also be taken by quite a number of people for insomnia and stuff. Do you think it really nicely supports the tryptophan supplements?
Do I think it supports tryptophan supplements? Is that what you're asking?
Yep. And it's the
It's derived from serotonin, which is derived from tryptophan. So in that sense, if you're supplementing melatonin and you don't have to make the melatonin, it may be helping to deal with the tryptophan deficiency problem.
Right.
I do worry about it because it does have such an important, such an important molecule in the blood indicating the wake-sleep cycle. So I wonder about if you're taking it in excessive amounts, are you disturbing what the pineal gland would normally do to control your wake-sleep cycle? I don't know. I know lots of people who are taking melatonin, even at surprisingly high doses. Personally, I would be a little concerned about that. And I don't know, some of you may have experience with that as well.
Sure. And what do you estimates about the possibility of developing these severe neurological disorders?
About the what?
About developing the severe neurological disorders?
What about it?
What do you think with the severity, how often it will develop with the long COVID symptoms?
How often, how much, those
Yeah,
kinds
yeah,
of things.
yeah,
It looks
yeah.
like quite a few
Just to check.
people are developing, brain fog and cognitive problems. So Parkinson's, of course, is a big one. And I believe that the virus is going to increase the prevalence of Parkinson's. And we'll have to wait and see because that takes time to develop. But it looks to me like it's kind of a good setup for Parkinson's disease. So that's the one I would most worry about. But also the prion disease itself, which is
Yeah.
very, very rare. But I think all of those and Alzheimer's, dementia, I think all of those are problematic. ALS, because they're all connected to misfolded proteins and the prion-like aspects of the spike protein, and the fact that it can get into the brain are all very disturbing through these exosomes. I think that to me is a very worrisome aspect. And I think that if people have a healthy immune system, they can, of course, keep those senescent cells at bay. They can clear those senescent cells with a healthy immune system. But many people do not have a healthy immune system. And I attribute that in part to glyphosate. That's something that I wrote about. I have a whole chapter on how glyphosate disrupts the immune system in my book. And of course, there are other chemicals that we're exposed to as well. So I think it's really important to maintain a healthy immune system. And I think one way to do that also is to get out in the sunlight. I'm a big fan of sunlight exposure because it's free, it's readily available, and it increases your vitamin D, helps to strengthen your immune system.
Yeah, and you have beautifully shown quite a number of nutraceuticals compounds like quercetin and all. The same question which I have asked previous speakers as well. Do you recommend any particular concentration which you reckon would be the best for those kind of compounds for the long COVID, or
I
it just
can't do that.
varies?
I don't treat people. I have no clue actually. So that would have to
Right.
defer to the experts
Sure.
on
No,
that.
no, absolutely. Thank you.
Yes.
Thanks, Dr. Seneff. Yeah, Dr. Chetty.
Stephanie, just to reiterate what you've just said, it's the infection with SARS-CoV-2 that will cause the syncytia formation and drive cells towards senescence. And those senescent cells, which should be eliminated by the body, produce a whole lot of toxic chemicals which drive inflammation.
That's right.
That's one
Perfect.
process that we're looking at. And then the second is with the spike protein acting as a prion-like protein on its own and causing damage, neuronal damage, as well driving alpha-synuclein production and causing aggregation, and in that way causing neuronal damage, as well causing the spread via exosomes, worsening that nerve damage. So
Mm-hmm.
those are the two mechanisms that we have at play. You mentioned the persistence of viral antigen in the gut itself. Is that due to bacteriophage activity that spike protein persists? Why do we have this increase in antigen for this length of time in the gut itself?
A great
Has
question.
they shown persistent viral infection in the gut?
Right. I know those are big questions and I don't have answers. I think it's all very interesting hypotheses and you probably know better than I do exactly. You probably have a stronger arguments for your hypotheses, but it looks to me like I think there's a possibility that you mentioned, it was mentioned in an earlier talk about bacteriophage like capability of the SARS-CoV-2 virus, that it can infect the bacteria and the bacteria can acquire DNA, coming from a viral infection. So it's conceivable to me that the bacteria themselves may have gotten some parts of the proteins in the virus incorporated into their own DNA, and now they're just producing those proteins. It's not a viral infection. It's just that the bacteria are expressing the proteins that came from the virus, segments of the spike protein, for example, that could be causing all this irritation and all these problems. It's possible. So I don't know. I think either continued infection hiding in the gut, or somehow some cells able to hang on to the spike. I don't know. I think it has to be some kind of continual production of the proteins of the virus, either by these bacteria that have acquired the genes, or by continued infection of the virus, hiding in, for example, senescent cells and sharing it, keeping it going. I don't know.
Can I ask a question on behalf of the audience? I thought this was an interesting one. What are the chances that long COVID is an autoimmune disease?
Yeah, I mean, that's certainly a possibility, because the spike protein, for example, has many, many segments in it that are similar to segments in human cells, human proteins that are produced by human cells. And so there are people who have shown that there's many, many different autoimmune diseases associated with certain specific proteins that have some sequences that are similar. So if you have antibodies to the spike protein, the whole thing could be driven by the antibodies without even having a continued infection. And that is a very interesting idea, that because of the overactive antibody response to the virus that's now sustained and is now attacking human tissues through molecular mimicry. That's quite a possibility. I think that we should consider that. There may not be any continued infection. It might just be antibodies. Excellent question.